Hi everyone, welcome to Myp Biology Unit 10 Consequences Part 4. Today we're going to talk about genetic engineering and cloning. Have you ever heard about genetic engineering? Of course, it's all where and it's actually ubiquitous. We're all the time hearing about genetic engineering. What is actually genetic engineering? Living things naturally create useful products. Genetic engineering can be used to make living things produce other more valuable products. For example, yeast naturally converts sugar into carbon dioxide and alcohol and is used in baking and brewing. Yeast can also be genetically engineered to produce vaccines for human disease. Genetic engineering is about changing the DNA of a living thing to change its characteristics. Sounds interesting, isn't it? Now, genetic engineering involve four main stages. Now, the stage 1, select the product or characteristic needed. For example, I want to cure hepatitis B, so I need an antigen for hepatitis B. Isolate genes from the specialist cells. So from the hepatitis B virus, I just actually isolate antigen for hepatitis B. Insert the genes in the target cell. So I insert the antigen for hepatitis B into the yeast and replicate the new organism. So they culture the yeast with the fermenters and I will get actually the hepatitis B vaccination. Now, GM crops. What is a GM crops? As I said, they are genetically modified. What characteristics might be useful in crops? Pest resistance, frost resistance, disease resistance, herbicide resistance, drought resistance, and longer shelf life. You know that because of the overpopulations, we are in danger, we are threatening by just lack of food. So the scientists have developed genetically modified products and crops that they are very tolerant to the frost, disease, and pest, herbicides. So by this way, they are no longer ruined by different environmental factors, and they are really all of them become a good product to sell in the markets. Now, types of GM crops. We have got herbicide resistant crops, we've got pest resistant crops, that this include insect resistance crop that it actually produce bacterial toxins that kills a specific pest. There's a disease resistant crop and crops with increased nutritional value. This includes, for example, golden rice. Now look at this. Potatoes can be genetically modified, so they are toxic to pests such as the Colorado beetle. You know, the Colorado beetle is the #1 enemy for the potato. So the gene for a powerful bacterial toxin is added to the potato plant. And if the beetle tries to eat the potato plant, it's killed by the toxin. What benefits might this have for the environment? Like frost-resistant crops, crops can be genetically modified so they are resistant to the adverse environmental conditions. For example, lettuces could be genetically modified to be resistant to frost. So GM lettuce and non-GM lettuce, you see the differences. Why are some people against the development and use of GM crops? What do you think about that? Plants with extra vitamins, so golden rice, as was said, that it has a higher value of the nutrition. So the golden rice is being developed to help fight vitamin A deficiency and blindness in developing countries. So early genetic engineering. People have been doing a simple form of genetic engineering for thousands of years. This is called selective breeding. Selective breeding or artificial selection is a process where people try and improve plants and animals by selecting and breeding only those that have desirable characteristics. For example, a farmer might choose 2 largest cattle in his herd and breed them together so that the offspring will be even bigger and produce more meat. Or breeding sheep to produce more wool, breeding wheat to produce more grain, and breeding tomatoes to become tastier. Other examples include breeding racehorses to become much faster, breeding dogs to obtain unique characteristics such as bulldog, greyhound, chihuahua. The genetic engineering in medicine. How can animals be genetically engineered to help humans? What is transgenics? Foreign DNA, including DNA from humans, can be inserted into animals. This is called transgenics. The protein encoded by the DNA can then be produced in the specific tissue of the transgenic animal at a specific time. This method produces a higher level of antibody more easily and cheaply than by using genetically engineered bacteria or mammalian cells. Transgenic goats. Look at this. For example, the gene for a human antibody can be introduced in the goats. Additional controlling DNA is also introduced, so the human antibody is only produced in the goat's mammary gland at a certain time. The antibody is then expressed in the goat's milk, where it can be purified and can be used to treat disease. Which came first? The egg of this transgenic chicken contained a human antibody that could one day help to treat cancer. What advantages does this method of producing antibodies have? Do you think that it's right for animals to be genetically engineered to help treat human disease? We're going to actually talk about this one. A new baby. Now look at this one. This boy got half of his genes from his mother and half from his father. It's completely random which ones he received. He may have his parents' best characteristics or the reverse. Are there any characteristics you wouldn't want your children to inherit? Of course. Sally has breathing difficulties. Her genes gave her cystic fibrosis. She risked repeated chest infection and lung damage. Molly could be fine, but one of her genes put her at risk. She has a high chance of getting breast cancer. Some women with the gene choose to have their breasts removed. What is a gene therapy? Children with faulty immune system have been cured by adding genes to their bone marrow cells. This is called gene therapy. A healthy version of the faulty gene is cut from the normal DNA and copied. The gene is added to harmless virus. The virus carries the gene into patient's cells where the healthy gene is released. The patient's cells can then make the correct product of the gene. The patient is then cured. Here we have got the cloning. What is it cloning? Cloning is the production of identical copies of organisms, cells, or DNA. A clone is a genetically identical organism or a group of genetically identical cells derived from a single parent. Cloning occurs in nature as a part of growth and reproduction. Now, why clone organism? Scientists can use genetic technologies to clone the whole organism. This is known as reproductive cloning. Farm animals, crops, plants with desirable features can be cloned, ensuring a constant food supply and high productivity. It is so that reproductive cloning could also help to increase the number of rare or endangered species. Natural clones in plants. Some plants are able to naturally produce genetically identical offspring, and the adult plant cells are called totipotent. That means that each cell has the capacity to regenerate the entire plant. The English elm can reproduce by vegetative propagation, a sexual reproduction that involves producing new plants that from existing the vegetative structure. If a tree that cannot withstand its current environmental condition, it still has a chance of survival because the basal sprouts can allow the organism to regrow matures from the original tree. Large-scale cloning of the plants, as you see in the picture. A plant with a highly desirable characteristics that produce many more plants with exactly the same genetic composition. The production of plant clones can occur at any time of the year. Disadvantages of reproductive cloning is that clones will be equally vulnerable to disease or pests, causing disease to spread very fast, affecting the whole population. And as you know, that because they have the same traits. Now, look at disadvantages of reproductive cloning. 1. The first cloned mammal, Dolly the sheep, died prematurely due to the lung disease. This raised concerns about the health and life expectancy of cloned animals. It is so that some cloned mammals will have shorter telomeres than other animals of the same gain. Telomeres are a piece of non-coding DNA that prevent the chromosome from degrading. They shorten as cells divide and are therefore considered a measure of aging in the cells. So here we have got the glossary of the new terminology of this lesson. So thanks for watching. Have a nice day. Bye-bye.